Specialized cells are cells that are designed to carry out specific functions in multicellular organisms. Although all cells share basic features, specialized cells differ in their shape, size, and structure to help them perform their roles more efficiently. For example, red blood cells are shaped to carry oxygen, nerve cells are adapted to transmit messages, and plant root hair cells help absorb water and minerals. By having different cells perform different tasks, organisms can function more efficiently. Understanding specialized cells shows how living organisms rely on teamwork at the cellular level to survive and grow.
The first specialized cell that we chose from the digestive system is the surface mucous cells. Surface mucous cells are specialized epithelial cells lining the inner surface of the stomach and the gastric peaks. This slide shows the labeled surface of mucous cell. The nucleus, located near the base, controls the activities of the cell. The rough endoplasmic reticulum and Golgi apparatus are responsible for producing and packaging secretion. At the apical region, there are mucous granules that stores the mucus before it is released into the stomach lumen. These structural features indicate that the cell is specialized for secretion. Next, surface mucous cells have several structural specialization that allows them to protect the stomach lining. Firstly, their tall columnar shape helps form a continuous and tightly packed protective lining. Secondly, the apical region contains mucus-filled granules that stores and release large amount of protective mucus. They also have a well-developed rough endoplasmic reticulum and Golgi apparatus, which enable continuous production and secretion of mucus and bicarbonate. Tight junction between adjacent cells prevent acid from leaking between them, while the bicarbonate in the mucus neutralize acids at the stomach surface. Lastly, rapid cells turnover allows damaged cells to be quickly replaced, maintaining constant protection of the stomach lining. Together, this specialization makes surface mucous cells the primary protective cells of the stomach. Next, I will pass to the next presenter.
I'm Elisa Sofia Junik binti Abdullah, and I'll be presenting about the structural specialization of parietal cells. Parietal cells are located in the lining of the stomach, specifically within the gastric glands of the fundus and body regions. The first one is pyramidal shape, where it allows the packing of organelles needed for secretion. The cell structure is adapted so it can tightly organize and fit many organelles required for secretion, especially mitochondria. The second one is extensive mitochondria, where it supplies ATP for energy-intensive or known as protein pump. The third one is tubulovascular system, where it stores protein pump and fuses with membrane when secretion is stimulated. This system is a specialized membrane network in the cytoplasm. The fourth one is microvilli on canicular membrane, where it increases surface area when proton pumps are emitted. And last but not least is the secretory canaliculi, where it increases surface area for acid secretion. For this secretory canaliculi, it has two states, where it is resting state, where the canaliculi is lined with microvilli and supported by actin microfilaments, and secretory state, where the tubulovascles are fused with the caniculary membrane, which increases the area and lengthening the microville for maximal acid release. Thank you.
Next specialized cell in the digestive system of the body is the Schiff cell. Schiff cells are specialized cells found in the gastric glands of the stomach and play an important role in protein digestion. Structurally, Schiff cells contain a large amount of rough endoplasmic reticulum. This adaptation allows the cell to synthesize large quantities of protein-based enzymes efficiently. In addition, Schiff cells have a well-developed Golgi apparatus, which modifies and packages these enzymes before secretion. The enzymes are stored in specialized secretory vesicles called zymogen granules. The zymogen granules contain pepsinogen, an inactive precursor enzyme. Releasing pepsinogen instead of active pepsin is a key specialization that protects the Schiff cell and stomach lining from self-digestion. Once pepsinogen reaches the acidic environment of the stomach, it is converted into active pepsin, which breaks down proteins into smaller peptides. Overall, the structural specializations of Schiff cells directly support their function in safe and efficient protein digestion, contributing to normal digestive homeostasis.
Well, hello, let me introduce myself first. So, my name is Andresiga Anwar and my matrix number is 107833. So, by today, I will present about interaction among cells, which is parietal cell, Schiff cell, and also mucous cell. So, this cell, it allows your stomach to digest tough proteins while preventing it from digesting itself. It also can be found within the gastric gland of the stomach lining. So, one of the example of interaction among cells, which is the secretory role. So, parietal cell, it secretes hydrochloric acid and intrinsic factor. With this, it can lower stomach pH level. Other than that, Schiff cell, it secretes pepsinogen, an inactive coenzyme, and also secrete gastric lipase for fat digestion. Lastly, mucous cell. Surface mucous cells secrete thick and also insoluble mucus. Then, he has mucous neck cell, which secrete a thinner and also soluble mucus. I think that's all for me. Thank you for your time.
Now, we will see how cells interact with each other. Step 1. Signal production. A cell produces a signal such as a hormone, neurotransmitter, or chemical messenger. Step 2. Signal release. The signal is released from the cell into the surrounding environment or bloodstream. Step 3. Signal reception. The target cell detects the signal using specific receptors on its surface or inside the cell. Step 4. Signal binding. The signal binds to its matching receptor, ensuring only specific cells respond. Step 5. Signal transduction. The binding triggers internal reactions inside the target cell. Step 6. Cellular response. The cell responds by changing activity such as growing, dividing, secreting substances, or activating genes. And the last step, coordination between cells. Multiple cells respond together, allowing tissues and organs to function in a coordinated way.
Hello, my name is Elvi Carlson Anaxon Hex bearing the matrix number 108-61. Today, I'm going to present on how the cells help to maintain homeostasis, specifically mucous cells, parietal cells, and Schiff cells in the digestive system. First, mucous cells secrete mucus and bicarbonate. These substances form a protective layer that prevents stomach acid and digestive enzymes from damaging the stomach lining. Next, parietal cells release hydrochloric acid, which creates an acidic environment needed for digestion and helps to kill harmful microorganisms. They also produce intrinsic factor, which is important for vitamin B12 absorption. Lastly, Schiff cells secretes pepsinogen, an inactive enzyme. When it comes into contact with stomach acid, it is activated into pepsin, which then will digest protein. Together, these specialized cells ensure effective digestion while protecting the stomach lining, helping maintain the overall homeostasis. That's all from me. Thank you.